Hello, everyone, and welcome to today's keynote presentation titled The Clinical Progression to Optimal Outcomes for Beta-Lactam Antibiotics, from Prolonging Infusions to Precision Dosing. This webinar is a part of the 7th annual event in the Drug Discovery and Development Virtual Event Series. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now, before we begin, I would like to remind everyone that today's event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located on the left of your screen. Now, if you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. And finally, as a reminder, today's presentation is educational, and it does offer free continuing education credits. Simply click on the continuing education credits image located at the right of your screen and follow the process to obtain your credits. Now let's get started. I do want to now present today's speaker, Marc Scheetz, Associate Dean of Research and Professor, College of Pharmacy, Professor, College of Graduate Studies, Departments of Pharmacology and Biomedical Sciences, Director, Pharmacometric Center of Excellence, Midwestern University. And for a complete biography on our speaker, please find it on the left of your screen. Marc, welcome. You may now begin your presentation.
Thank you so much for that kind introduction. Any questions after this presentation, you're always welcome to follow up with me. You can find me on Twitter or via email at our website here. So today I'd like to go through the objectives. We're going to talk about the PK/PD, the pharmacokinetics and pharmacodynamics for beta-lactams. We're going to describe the variability of beta-lactam exposures. We're going to list and describe the strategies to achieve these target goals that we'll go over. And then finally, we're going to review the recent clinical literature on these strategy outcomes. Really, what is out there right now in the literature, how is that supported, and where should we go from there? So the first objective here is to review the pharmacokinetics and pharmacodynamics for beta-lactams. And I know that this is a broad and generally interested audience, so I'll start from a little bit of the beginning. So pharmacodynamics are really essentially what is occurring at the level of the antibiotic with that bacteria when we're talking about bacteriology. And what I'm showing for you here is a simplified cartoon of how the pharmacodynamics work and how in the laboratory we determine how that antibiotic bacterial interface comes out quantitatively. So what you do is you take a series of test tubes and you put some, what essentially is a modified Kool-Aid that helps the bacteria grow, and you fill those all the way up to the top. Then what you do is you take antibiotic and you put those into each of those test tubes. And what we do is we call a serial dilution, and the most common one is a doubling dilution. So you can see all the way at the right of the screen, we start out at 4 milligrams per liter. And then we serially dilute those down 2 milligrams per liter, 1, 0.5, and then a no antibiotic control. After that, what happens is we take a set amount of bacteria, say something like 10 to the 4th of bacteria, so colony-forming units, and we put that into each one of these test tubes. So that goes into each one of these test tubes. Then we put those test tubes into a nice warm environment for around 18 to 24 hours, and we see what happens. Now, the solution is set up for the bacteria to grow well. So what happens is the bacteria will grow quite well if we have not inhibited it with any antibiotic. So in this example, what we're seeing is the blue represents turbid. That means the bacteria really flourished. They took off, they grew. If you were to look at this, it would be a very milky white color and cloudy and difficult to see through. And you can see that in this case, the antibiotic did that for 0, 0.5, and 1 milligrams per liter. Now, the antibiotic did start to work at 2 milligrams per liter and 4 milligrams per liter. We call that first test tube where the antibiotic prevents that turbid growth, and you can actually see clearly through the test tube as the minimal inhibitory concentration. And so then what we can do is we can plate those out. We can take that solution, we can plate it out. We know that just because a solution is clear doesn't mean that it's free of bacteria, right? Everybody that has ever gone hiking in the woods has always been told don't drink the stream water, because even though it may be clear, there's still very much a chance of bacteria being in there. So at the minimum inhibitory concentration, that does not mean that you have killed all the bacteria. It just means that you have visually inhibited growth, and you can plate those out and you can see those bacteria. Now, the minimum bactericidal concentration is a 99.9% kill from your starting initial bacterial inoculum. The MIC is the pharmacodynamic endpoint that's really been linked in the literature over the course of time to outcomes. So that's the one that we're going to focus on in this lecture. Okay, so now we know how we determine MICs. We can start to talk about how we can start to assess the pharmacokinetics of antibiotics, and then that interface with the PD, how it interacts with the bacteria. There are multiple models that are set up. I'm showing you one of the more complex and more standardly used in vitro models, and that's the hollow fiber infection model. And what you can appreciate here is that you have a couple separate bottles, and they all get driven by a pump, and they end up here in this actual hollow fiber system. Now that blowout there that we're seeing, that's a cross-cut section of the hollow fiber system. And what you can see is that the hollow fibers are shown in those yellow circles, and then there is this extra capillary space where the bacteria can grow. So the bacteria get put into the system, and this is a slightly better representation of just having a static system where you would just put bacteria in a test tube, because it really allows these multiple compartments, and it allows the bacteria to see things such as changing antibiotic concentration. So you see there is another pump all the way at the bottom left corner here. And by changing the rates of these pumps, you can set it up such that the concentration of bacteria in the hollow fiber system mimics what you would see in the body. When we start moving into the in vivo systems, the mouse is one of the most commonly utilized infection model. And what I'm showing here is the murine thigh infection model. Generally, what's done here is that the mice are rendered immunocompromised, and that is mainly to look at the pure effect of the antibiotic on the bacteria and take the immune system out of the equation. The other thing that you'll see that is done is generally uranyl nitrate is added, or some way to inhibit the kidneys, essentially kill the kidneys, so that the antibiotic is cleared more slowly. Now, because mice are smaller order animals, they clear much more quickly according to the rules of allometry. And so we have to end up inhibiting the kidney so the antibiotic can stick around long enough. Those mice then get infected with a certain number of bacteria, say 10 to the 7th colony forming units per mil. And then after that, you can then look and see what the outcomes are in the mice. Okay, so that is how we're looking at pharmacodynamics. The pharmacokinetics is really what the body is doing to the drug. And I'm showing you a concentration time profile here. So you give an antibiotic, and you can appreciate that if you give it as a bolus dose, you get a quick rise and it goes up. And then we will see what is called a Cmax. That's what's shown in that first bar there. And then the antibiotic will be cleared by the body. And we see that concentration going down, 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 down until it is the end of that, almost looking like a triangle. Now, we just talked about the MIC. So the MIC is a concentration, and that can be plotted on this concentration time curve. And you can draw that as what I'm showing here as an orange dotted line across there. Now, with all antibiotics, we classify them as either being dependent on getting high maximum concentrations, total exposure concentrations, or some amount of time-dependent action, which is shown as the time over the MIC, which is shown as that horizontal bar going across the bottom there. And what you can appreciate here is about 50% in this case, that antibiotic concentration remains above the MIC. Okay, now let's talk specifically about the beta-lactam targets. So for the beta-lactam targets, there's been quite a bit of research. A lot of the original research was done by Dr. Bill Craig. He's has many of the seminal publications in this area. And what you can see is that if you look at the various ways that you can classify the antibiotic pharmacokinetic to pharmacodynamic relationship, you'll see that in the top left-hand corner, the peak to MIC ratio for the beta-lactams does not do a very good job of explaining it, nor does the AUC to MIC ratio, but rather the time above the MIC is what explains the outcomes for beta-lactams. That is, you want the concentration at the site of infection to be above the MIC for the longest percentage of time. And when we look at the various beta-lactams, here's another nice graph from his very early publications. The penicillins are shown in circles there, and here I have it as highlighted as red. And you can see that the penicillins require slightly less time above MIC than do the cephalosporins that are shown in the triangles. And you can see that because the y-axis is the mortality after 4 days of therapy for mice here, and you can see that you get this very nice precipitous drop, and that number ends up being somewhere between about 40 to 50% here in that experimental model. And we'll talk more about what has happened in the more recent experiments. Now, there was a really nice meta-regression that was performed looking at all of these PK/PD targets by Sophie Dace, and what they found is that there's a lot of heterogeneity. There really is a lot of heterogeneity, and it was really difficult for that meta-regression to drill down on the variables that impact the outcome for beta-lactams. And I think the systematic review from this paper is probably the most instructive. It really gives us the most information. And what you can see is that you really need this time over MIC to be about 30 to 70% for most of our bacteria that you would encounter in clinical practice. And that's true for a variety of beta-lactams. Penicillins require a little bit less, carbapenems even less yet, and cephalosporins a little bit more, but somewhere in that range of 30 to 70%. Now, we'll talk a little bit later about the various ways that we can administer beta-lactams. And so for our prolonged infusions, that's shown in the upper right quadrant here, upper top right quadrant, you really want to stay above that MIC for 100% of the time, or as much as 4 times 100% of the time. And we'll talk about that in just a minute. Okay, so why does all this matter? This all matters because the outcomes do translate from our in vitro models to our in vivo models in mice, all the way to what we've seen in a lot of retrospective studies for clinical outcomes. These are some data that came out from our group, and what we're showing here is that for patients that had gram-negative bloodstream infections, and they were treated with the antibiotic cefepime, which is a cephalosporin, the outcomes were dependent on 2 things. One, how sick the patient was, and that's shown on the x-axis down on the x-axis of this graph. So we looked at this with the modified Apache 2 score. And on the y-axis, what we're looking at is the probability that the patient lives through the hospital course, so the probability that the patient survives. Now, the 2 different lines here that are shown by the filled in red circle, which is achieved their 68% time greater than MIC. Did not achieve 68% time over MIC is in the red, and they did achieve it in the blue, which is the open blue circle, greater than 68%. So if they achieved a time over MIC of 68%, they're shown in the blue. Now, what you can appreciate here is that something that we know very well, that the sicker a patient is, the the less likely they are to survive. So as you move to the right along that x-axis, you can see that these patients are indeed much less likely to survive as that goes on. The other thing that we can see is that it is important to achieve that target, that time over MIC of 68%. So for the patients that did, there is a separation between those two lines and the confidence intervals of those two lines. So statistical significance is really defined when those two confidence intervals do not overlap. And what you can appreciate here is between the Apache scores of 9 and about 23 there, what we're actually seeing is that this is where achieving optimal pharmacokinetic, pharmacodynamic relationships is most likely to impact the patient end outcome. And that being a very relevant outcome, that being probability of living through their hospital course. And in this paper, we called it the Goldilocks effect. And I think it makes general intuitive sense. That is, if you're very healthy, it doesn't matter if you have optimal antibiotic exposures because you're going to do well anyway. You're very healthy and you really don't need the antibiotic nearly as much. On the flip side, if you are extremely sick, an antibiotic also will not help you. So antibiotics, remember, do not raise the dead. Rather, they are very useful tools and they help us achieve our optimal outcomes. But if your Apache score is into the 30s, you really have such a pre-treatment prognosis that's bad that an antibiotic is much less likely to help you. So what we found in this study is that there is this Goldilocks range of being sick enough, so having an Apache score of about 9 to 23 here, and achieving your optimal antibiotic exposure in this range is what predicted the best outcomes for patients. Okay, so that's the backdrop that we're talking about for optimal PK/PD. The next thing that we hear very often is that beta-lactams are very safe drugs and there's not a lot of variability in our beta-lactam exposure. So let's explore that. The first thing I want to show you here is a nice slide that Dr. Jim Rose was nice enough to share with me. And what he did here is he compared the pharmacokinetics of that were obtained in critically ill patients to those that are reported in the package insert. Now, remembering that in the package insert, patients are generally much healthier. It's much more of a select group and a very narrow patient population that are used in the pre-approval stage of those drugs. When we look at our critically ill patients, they represent the patients that our clinicians are seeing every day. They are very different generally from most of the registrational trials, and really the way that we need to think about using the drugs in our own patient populations. So when we look at the difference between clearance calculated among these very common beta-lactams, piperacillin, cefepime, imipenem, meropenem, doripenem, you can see that we're seeing clearance differences of 148% and 47%. So there's this very large gap between what is reported in the package insert and that which we're seeing in our ICU patients. Now, it's very common for me to hear people say, well, you know, Mark, I know this. I mean, we know that the ICU patient population is very different from the package insert population, and that's to be expected. But I think what's really not appreciated nearly as well is how different it actually is. So one of our former postdocs put this together as a review article, and I think it really nicely illustrates in just one beta-lactam how much variability we can see. So if you look at the very bottom, this is cefepime, and we're looking at the clearance of cefepime that's shown in the x-axis in healthy people at the very bottom, and that's shown in blue. And you can see that there's a very narrow range of cefepime clearances in that blue healthy range. As we go up one, we see that the renally impaired patients, as expected, shift to the left. So they have less cefepime clearance. So renal impairment should predict that cefepime clearance goes down because cefepime is a very highly cleared antibiotic by the kidneys. When we look at our relevant patient populations, we start to see a lot of variability. So when we look at febrile neutropenia, which is shown in the purple upside down triangles, we see very wide variability. In our pneumonia patients, we see various authors have found very different clearances. So clearances that are different by as much as threefold. And then the same is shown in our septic, burn, and critically ill patient population. So very, very large distributions of clearance in these patient populations that actually require treatment, not the healthy patients, but those that we wish to use cefepime for. All right, so I often hear that, yes, we know that there is variability and it's probably this very nice Gaussian distribution. And we can simply take a look at these things and then we can make our predictions based on these Gaussian distributions. And so what I'm showing you on the left is a nice Gaussian distribution. You give a drug and that's what you would expect to see for distribution of a single concentration that is drawn. And if we look at multiple concentrations over time, what you generally see in the publications that you're often reading are these nice box plots, and they look extremely pretty. Well, what I overlaid here are what would be the individual patient samples that you would expect to see. And again, you'll probably say the same thing. That looks really pretty. The box plot did a very nice job of capturing that. And the reason they did the box plot is to just simplify it for us. But what I'll submit to you is that while we see those individual concentrations, what we tend to see is if we look at the individuals that make up those individual data points, that they tend to overlap. And that's because we tend to see a lot of covariance. We tend to see a covariance between our variables like clearance and volume of distribution. And so what I've shown you here is a spaghetti plot, and each patient represents a single strand of spaghetti on this concentration time plot. So this was ceftazidime, and that's shown, the ceftazidime concentration is shown on the y-axis, and the time is shown on the x-axis. And what you can appreciate is those pieces of spaghetti start where some pieces of spaghetti start really high, they get high concentrations, and they clear really quickly, whereas some start much lower and they clear really slowly. And so we have this covariance between clearance and volume of distribution. And if you ask pharmacokinetic modelers, they say, we know this. I mean, this is extremely common knowledge, and this is reported in all of our pharmacokinetic papers. But I think there's a disconnect between what is being explained and what I think is being appreciated at the clinical level. So we'll try to illustrate that for you. So what we tried to do in this case was to look at that myth that there is this beta-lactam variability that you can adequately describe with many of these clinical covariates. And if you ask a clinician, even a first-year trained clinician, how they should adjust their cefepime dosing, they will very clearly tell you that you can do that very well with cefepime, with creatinine clearance. You just have to know the patient's creatinine clearance. And once you know that creatinine clearance, you can then come up with a dose that should be very safe and effective for that patient. So let's take a look at some real world data. So for this study, we were very fortunate to work with our friends at the University of Florida, led by Chuck Peliquin and Mo Alshir and Veena Venugopalan. And what they had is they had data from 75 patients in the MICU that had received cefepime. So these are real patients in the ICU, and they were getting cefepime for the clinical course of their infection. My postdoc, Dr. Jack Chang, was the person that did the modeling for this study. And what you can see is that he fit a model, and in the lower left-hand corner here, you can see that the population predicted concentrations on the X compared to the observed concentrations on the Y are not very well described, with an R-square of 0.17. You might say that's because the model is very bad. However, these are real world data, and this is the best possible model that could be built in this case. You can see that the model did do a very good job at predicting the individual. That is, individuals are more likely to act like an individual than they are to act like a population mean. And we'll get into this just a little bit more. So for for this paper, what I really wanted to do was to give these illustrative examples of what we see as pharmacokinetic modelers, these things that we know to be true in pharmacokinetic modeling, and really, I think, don't make it out into the clinical realm nearly as well. And so what we did is we took that model and we used the covariance matrix and all the basic standard stuff that you do in PK/PD modeling. And we simulated 10 patients that had the exact same creatinine clearance. So from a clinician standpoint, this is a patient that has the same creatinine clearance. They should all get the same dose, and they should all have the same cefepime clearance. What you can see is that these patients, in fact, do not have the same cefepime clearance. They have the same creatinine clearance, but the cefepime clearance is, in fact, very different. And you can see that by the very different trajectories of how fast these concentrations drop, again, on that cefepime concentration time curve. And how much different is it? It actually was about 10 to 30-fold variable. So about 10 to 30-fold difference between the 5th and the 95th percentiles for our N of 10 and N of 1,000 sims respectively. So if you only simmed it 10 times, the 5th and the 95th percentiles vary tenfold. And then if you did it 1,000 times to simulate a much larger group, we saw as much as 30-fold variation between those percentiles. So 30-fold variation. And this is a number that I think you'll hear over and over again, or see over and over again if you review the pharmacokinetic, pharmacodynamic literature for ICU patients. Lots and lots and lots of variation. But then you might say, well, does it matter? I mean, there is this variation, and we know that we have creatinine clearance, and creatinine clearance varies highly in these ICU patients. And so we looked and we incorporated creatinine clearance into our model, and we looked at the relationship of creatinine clearance on the x-axis to cefepime clearance on the y-axis. And this is a 3D histogram where the number of patients goes up. So they stack up like buildings in the case that they share the same data point there. Now, what you can appreciate is that the middle group here, so if you run a lowest line here through the middle, no patient actually lives in there. So there's no relationship between creatinine clearance and cefepime clearance that we can predict using anything like a lowest line, or I'll show you some more ways you can look at it in a minute. And, when I started thinking about this, I started thinking back to my days when, you played the board game Battleship. And the whole idea of Battleship is that once you get a hit, once you predict something that is a hit, the next goal that you have is to move on a linear line to keep moving and then sink that person's battleship. So you know that these ships are very linear. And so then your next pick is going to be in a linear line. You're going to try to guess around until you get to that linear line. Once you get 2 in a row, you'll just keep going because the ship is really this linear relationship. But what you'll see here is that there is no linear line. So there is no linear line. Clinicians are using the x-axis, a patient's creatinine clearance, to try to predict cefepime clearance. And there's no rhyme or reason here that really allows them to do that. So simply knowing a value does not allow you to sink that patient's battleship, or that is, in this case, guess that patient's cefepime clearance. Now, I took that graph and I also just turned it into a 2D graph to make it perhaps a little bit more standardized, something that you're used to seeing. And again, on the x-axis is a creatinine clearance, and on the y-axis is cefepime clearance. And you can see that if you fit a linear fit, again, nobody really lives in that linear fit. And then really on the line of unity, where a creatinine clearance of 100 mils per minute is set to 100 mils per minute of cefepime clearance, nobody lives along the line of unity either. So what we're demonstrating here is that there is not a great relationship between cefepime clearance and creatinine clearance, that there's a lot of variability here. It is a good relationship. It's still probably the best covariate we have for describing cefepime clearance. However, there's much more variability than I think is generally appreciated. Now, what does this mean? Where does the rubber meet the road? We know that creatinine clearance is usually within about a 30% prediction. So if we were to take a patient that had a creatinine clearance of, say, 59, and that's shown in the tan, and actually it looks like it got converted over to red in this graph. And then we vary that up by 30%, so within the margin of error, and that becomes 76.7, and vary it down by 30%, it becomes 41.3. What we can see is that our probability of target attainment, shown on our y-axis, varies quite a bit across MIC. And so if you were treating a patient with a cefepime MIC of 2, and you thought the patient's creatinine clearance was 59, if in fact their creatinine clearance, their glomerular filtration was really 76.7, you can see that that really drops you down, and that's at the left edge of that yellow middle bar, down to below 80% probability of target attainment. So you might have thought that you were okay, which is one step up, that 90% probability of target attainment in the red/tan, but in reality, you're actually much lower than that. And then that gets amplified at 4 milligrams per liter, which is also considered to be susceptible dose dependent. All right, so now hopefully we've shown you that there is this very nice pharmacokinetic, pharmacodynamic relationship that can be had, and that these patients have quite a bit of variability. Now, what can you do about it? So what can you do about it? So one thing that you can do is to give a continuous infusion. The traditional infusion would look like the black line here. You give a 30 to 60 minute infusion, you get a nice peak up, and then it gets eliminated. You can do a prolonged infusion. Maybe you infuse it over 3 or 4 hours, and then you see what you see in the blue line, where you get this more gradual peak up, and then it eventually comes down when that infusion gets turned off. Or you can give a continuous infusion, which you just give that same amount of drug continuously, and you never shut off the drip, and that's shown in the red. And then you really have to think about the 2 different pathways that you're going to treat your patient. One is the population pathway, and this is the way that's done most commonly really around the world. And then the second way is to do individualized dosing. So using the patient's own data, and in this case, the patient's own levels, to come up with the best approach for that individual patient. We'll explore these. All right, so just showing you again how the various ways you can administer cefepime. So I just created a patient that was getting cefepime 2 grams that had volume of distribution of 30 liters and a clearance of 5.9 liters. If you give it as a 0.5 hour traditional infusion, what you see is in the blue. If you give it as a prolonged infusion, a 3-hour infusion, you see the green concentration time profile. In the tan is a 4-hour infusion, and then finally a continuous infusion in the red. So these are the ways that you can change your infusions, and in this patient that I simulated, what you would expect to see. But remember, we just spent a lot of time talking about patients not being means, rather they're individuals. So if we were to think of the patient as a mean, what we would see is in the green line. But remember, just go back to that 10 to 30-fold variation that we actually really expect. You cannot just assume that your patient will end up at the mean concentration, but rather they're more likely to be either a red or blue piece of spaghetti rather than that green giant piece of spaghetti. So what can we do? So in order to individualize therapy for these patients, what we can do is we can measure drug concentrations. And we'll spend just a little bit of time talking about measuring drug concentrations and then using models to understand which piece of spaghetti that patient actually is. We could have a whole separate talk on optimal sampling times. I'm not going to do that today, but the general goal is to figure out which piece of spaghetti the patient actually is. And you can appreciate that if you draw a concentration in the box here and you get the red amount, it's pretty easy to see that that patient's the top spaghetti. And if you get the blue amount, you can see that the patient's down there, a different spaghetti. The most frequent thing that we do is to measure trough concentrations, which is off here to the right. That's the purple. And then it's really difficult to discern which piece of spaghetti that patient actually is. So troughs are the least informative concentrations that we can draw if you're just drawing a single concentration, but we'll save most of that talk for a different time. For this time, just suffice it to say that the best way to understand what a patient's overall exposure is to actually measure. And then we can put them into some specific monitoring programs, and many of them use these Bayesian methodologies. I'm just showing you a quick screenshot from DoseMe for a patient that was receiving cefepime. And we'll get very quickly into what these Bayesian analyses can do. So the whole idea of a Bayesian analysis is really to use prior information, what's known about a drug, what's known about an overall population, use that in conjunction with a measured concentration, current information, and then use that to understand what the most likely scenario is that's in front of you. So let's just go through a very quick example. So you decide to go out and try a new pizza place, and you look at Yelp, and in Yelp you see that it has 92% correct orders. You also look through Yelp and you find out that, you know, when there were wrong pizza orders, management served the table 50% of the time, but management only serves 10% of the pizzas. So you walk into this pizza place and the manager walks out and says, "Oh gosh, we're so busy. Tonight I'll be taking your order." What's the probability that your pizza will be incorrect? So we can plug all of this information into our formula, and I'll leave this here if you want to go back and calculate it yourself. The probability of management given a wrong pizza is 50%. The probability of a wrong pizza from before was 8%, and the probability of management was 10%. So that's how the math works out. What that all works out to is the probability that you will end up with a wrong pizza in your individual visit is now 40% on the basis that you now know that a manager was taking your order. And I can let you go through the math here on your own. We'd be glad at some point to come back and have a more in-depth discussion here. But one thing that I'd like to point out, clinicians know the number positive predictive value very well. They know the definition of positive predictive value very well. And what you can see is that the Bayesian posterior concentration is the same as the positive predictive value. So this is using the known characteristics of the test and your own result to understand what the likelihood is. And so you'll see it's 40%, whether you call it the Bayesian posterior or you call it the positive predictive value. All right, let's finish up now and talk about what clinical literature exists and how it can guide you. So a little bit earlier this year, actually late last year, there was a nice study by Paul Williams and Jason Roberts, and they did this very nice large survey of 538 people, and they asked them, how often are you using therapeutic drug monitoring? How often are you getting concentrations for beta-lactams? And they found that it was really only 43% worldwide for Piptazo and 39% for meropenem, and that's shown in the bar graphs here. And you can compare that to other drugs that are more commonly have therapeutic drug monitoring, things like vancomycin and aminoglycosides, where we see those numbers at 90%, 82% for vanco and aminoglycosides, respectively. And in the U.S., I'm very sad to report that we're actually losing. So we're doing very poorly in the United States. So in the United States, our piperacillin and tazobactam monitoring was really only reported by 7% of respondents, and the same for those that are using meropenem. So only 7% of respondents had therapeutic drug monitoring and assays available for those two drugs. And 0% of our respondents were using any software that could help them calculate. So some software that can help identify which piece of spaghetti that patient is with even a single concentration. I think the good news, though, is that this was a previous survey. And in the previous survey, the numbers were even more dismal. So when this survey was conducted in 2015, what we saw is that there were only 10% of respondents that were that had piperacillin tazobactam being TZP over here, and 8% of respondents that had meropenem concentration monitoring available. So you can see that the numbers that we're looking at now are actually much, much higher than they were even just several years back. So I think that that bodes really well, that we're starting to see our clinicians have access to assays. And that has always been something that has been thought to be important. Across Europe, Australia, and many sites in Asia, many sites have more access to therapeutic drug monitoring than we do in the United States. Here was a French survey. In this French survey, 75% of French respondents had access to beta-lactam therapeutic drug monitoring. 60%, so 60 of those 75% actually had on-site beta-lactam assays, which is even better because you really need to be able to make these decisions very quickly. If you have to send your assays out to another site, it takes 4 or 5 days, and you're talking about a patient that was critically ill. Oftentimes, the horse is already out of the barn. So you needed those data many days sooner in order to make an informed decision for your paper. Now, what I found interesting from this French survey is so 70% didn't know if concentrations were reported as free or total. And that's important because it actually impacts what the actual number is. And so there's, they have the assay, but there perhaps is a knowledge gap in some of the clinicians that have access to these assays and thinking about what they can actually do with them. And with that, I'd like to talk about the barriers to beta-lactam precision dosing. I think this this study by Aaron Barreto is really nice, and it really sort of highlights that one finding from the French survey. And in this study, Aaron was able to recruit 38% physicians, 33% pharmacists, 15% of the physicians were fellows, and then advanced practice and 14%. And she has a really nice graphic in here that shows the many different things that impact knowledge base of what's going on in therapeutic drug monitoring programs for beta-lactams and beta-lactam assays. And you can remember just a minute or two back, we said that we thought that drug assays were, one of the big factors in being able to help personalize care for our patients. And in this study that Erin completed, the survey that she completed, she had 3 sites, and the 3 sites are one site that was just about ready to implement beta-lactam therapeutic drug monitoring and had the assays ready. One site that had partially implemented a therapeutic drug monitoring program and had been using these assays. And finally, a center that had fully implemented the assays and had a full therapeutic drug monitoring program. And Erin asked a bunch of questions that were really good. And I highlighted the fully implemented as green and the not implemented as red. And you can see that there's large gaps to the answers of these questions, like whether or not a clinician felt comfortable recommending beta-lactam TDM. Only 27% in the not implemented group versus 87% in the fully implemented group. If we go over to the barrier side on the right-hand side, clinician unfamiliarity was at 82% in the not implemented and 56% still in the fully implemented group. So even though the site had implemented beta-lactam therapeutic drug monitoring, many of the clinicians still felt that they had quite a bit of clinician unfamiliarity. And so I think what this suggested to Erin and what I took from her study is that it's just not the assays. So just getting access to assays is not in and of itself good enough. You actually have to go a step beyond that. You have to educate your clinicians and really have a support system for them. So not just giving them a one-time, here is your one-page information on how to do beta-lactam therapeutic drug monitoring, but rather having sort of a full service that's able to help them soup to nuts. And then so her conclusion was that it wasn't just assay variability, but rather lots of these other factors, and really needed to take a comprehensive approach to rolling these assays and therapeutic drug monitoring out. Okay, so you don't have beta-lactam assays. What can you do? And I kind of show this graph. I find it this picture, I think is quite funny. So these people are, you know, enjoying a nice drink while they're receiving their prolonged infusions. And with many of the IV access options now and the pumps, this actually is reality. So prolonged drug infusions are a lot easier than they were in the past. I'll highlight the international consensus recommendations for the use of prolonged infusion beta-lactams that just got published late last year. And I was fortunate to participate in that with Dr. Lisa Hong, who was the first author here. So I want to just hone in on two really quick questions. The first question being, is there a role for therapeutic drug monitoring of prolonged infusion beta-lactams? And after a very comprehensive literature search, you know, engaging 7 different international groups, we said that we cannot recommend for or against routine TDM for prolonged infusion beta-lactams at this time. And I will have you look forward to the future. Dr. Barreto and Dr. Jim Rhodes are going to be leading the international consensus recommendations on the role of therapeutic drug monitoring for prolonged infusion beta-lactams in the future. At the time that we reviewed the data, there were 3 randomized controlled trials, and I'll fast forward to show you what they showed. What you'll see is that they had numbers enrolled of 41, 32, and 38. So very, very small enrolling trials. So we applaud them for getting the trials going, but these are very, very small trials. And of course, with numbers like this, we're not surprised that there was really no difference in outcomes between those that had used TDM and those that had not. So the next question that I wanted to quickly highlight is, should prolonged infusion beta-lactams be preferred over standard infusion in severely ill adult patients? And we suggested that that's true, that you should use prolonged infusion beta-lactams in your severely ill patient population, and that was a vote 17-0 in favor. When we look at sort of the funnel plot here, and we look at the overall odds ratio, risk ratio, you can see that there's a slight hint that prolonged infusion, which is shown on the right, is better from all of the studies that had looked at prolonged infusion versus short infusion, and short infusion shown on the left. Just really quickly highlight some other things from that from that consensus document. The preclinical targets still said to shoot 40 to 70% for our standard infusions of time over the free drug concentration of the MIC. So free drug concentration over the MIC. And then if you're doing a prolonged infusion, to aim to be over that MIC at a free drug concentration the entire time, and up to 4 to 8 times higher than that MIC concentration as a free concentration. We saw equivalent or better killing for prolonged infusion compared to standard infusion. We didn't really see any additional harms that were identified. We already went over recommendation 4 and recommendation 7. The other thing I'll point out is that the clinical data tended to mirror the preclinical data. That is, that the clinical data tended to come up with the same numbers that we saw preclinically. So 50 to 70% free time over MIC. And then for our prolonged infusions, again, trying to hit at least 100% free time over MIC, and maybe up to as much as 4 times that value. All right, so I'll finish up, and we're just about coming to time. You should look forward to the BLING-3 study that's being led by Dr. Jason Roberts. That study was completed in June of last year, and they've enrolled over 7,200 patients, and they have 170 patients that were recruited for the PK/PD sub-study. This is going to be presented at a critical care conference in Belfast this coming summer. So look forward to that. We should see results of that RCT of 7,000 patients of continuous infusion versus intermittent infusion. Now, what you might say is that, Mark, I know there's already a randomized controlled trial out there of continuous versus intermittent infusion meropenem, and that was not covered by your international consensus document. So let's take a quick look at this. That is absolutely true. We did not cover it because its results came out after we had published the international consensus recommendation. So this is 607 patients, 303 randomized to continuous administration and 304 to intermittent. And if you look all the way to the right, there's a whole lot of nothing going on, really no differences in their individual or composite outcomes, no difference between continuous infusion and intermittent infusion meropenem for patients with sepsis. And I really applaud the authors here. We tried to write a letter to the editor, JAMA, to really get a discussion going, but there's a lot of questions about this trial, and it really just comes down to just needing to get enough subjects. And this, even though they enrolled 600, which is a feat, I mean, it's absolutely difficult to enroll 600 subjects. Of those 600 subjects, 20% had pathogens that were resistant to carbapenems. 30% had no pathogen, 36% had gram positives that were not thought to really benefit from any sort of meropenem strategy. And so that's 86% of patients where we really don't expect them to have benefit. And then the question is who can benefit? I think the answer is those that have an elevated MIC, those that are the Goldilocks sick, not too sick, not too little sick, and those that have altered PK, this variability that exists. And so I did a really quick sample size calculation, and you can see that if we're trying to drop from a 50% outcome to see a 5% difference, so 45% to 50%, we need over 3,000 patients that are in that Goldilocks window to really see a difference. So I'm really hopeful that the BLING trial will give us some good insight there. They have the chance at looking at clinically meaningful differences. This first study was a very great step in the right direction, but I don't think that we can take no benefit as no ever benefit. Rather, it was just not seen in a patient population that was not expected to benefit in the first place. So the key takeaways here are that beta-lactam pharmacokinetics are highly variable. We see this really amplified in the ICU patient population. I think we really need to start thinking about our patients as individuals rather than these population means. And if you don't have the ability to treat your patients as individuals, and using prolonged infusions for beta-lactams is probably the best that you can do to getting patients the best chance of success with their antibiotic. Therapeutic drug monitoring is not common right now in the United States. It's more common elsewhere, and hopefully it will be more common in the future. And we'd love to see you sort of pick up that flag and help that happen. I think the new studies that will be coming out hopefully will show that this is in fact can result in positive outcomes. And with that, I want to thank all my collaborators, all of our postdocs who have helped generate so much of this work. And after that, happy to move on to questions. Unfortunately, I'm traveling during this event, but we'll be glad to take questions at a follow-up time. So thank you to LabRoots for having me. I very much appreciated the chance to talk to this group here today. Thank you.
A big thank you again to Marc Scheetz for his time today and his important research. And before we go, I'd like to thank the audience for joining us today. Questions that were submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand for 1 year, and that's through February 21st, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you so much for joining us. Everyone have a great day. Bye-bye.